Hello, going through the first 20 questions of the IB20 trial paper 1. So question 1 is just asking you to calculate the actual size, although it's just an approximation. So we just want to remember the equation I am. We want the actual size, so we're looking for image over magnification. Now if you measure that diameter, let's say it's around about 15 millimeters, so actual size is going to equal 15,000, because it's obviously going to be in micrometers, divided by 2,000, which gives you 7.5 micrometers. So then you look at the possible options, and the maximum diameter then is A, 10 micrometers. In question 2, you're asked what's going on in this question. All of the chromosomes here are aligned in the middle metaphase, so it's C metaphase. Question 3, which processes are involved in the development of cancer? Well, mutations occur in oncogenes, that is correct. Oncogenes prevent cancer, that is not correct. And oncogenes affect the cell cycle regulatory proteins, that is correct. So we're looking for 1 and 3, so B. In question 4, we're looking at the movement of potassium ions and sodium ions. Now, if we have a cell, at the moment, we've got lots of potassium on the inside and lots of sodium on the outside. So if this was passive diffusion, we would expect potassium to go out and sodium to come in. So we look at this, each statement. Potassiums diffuse in, nope. Sodium ions diffuse in, and potassium ions diffuse out. Now it might make sense for that to be the case, but the question is saying what explains these concentrations, i.e. how would these form? Now if potassium is diffusing out, then actually this number would be getting lower and that number would be getting higher. So that doesn't explain the formation of these particular numbers. Active transport pumps sodium ions in, well that doesn't work because it's going with the concentration, going not against it. Active transport pumps sodium ions out, yes, because it's going from low to high, and that would explain those numbers. And potassium ions in, yes, going from low to high, and that would explain those numbers. So the answer is D. In question 5, where do hydrogen bonds form? Between a slightly negative charge of hydrogen, no, hydrogen's positive. Between a slightly positive charge of hydrogen and a slightly negative charge of oxygen within a water molecule, no, not within. Between a positive charge of hydrogen and a negative charge of oxygen in different molecules, yes. Let's just check. Negative charge of hydrogen, no, so the answer must be C. In question 6, this is a factual recall question. So we learn that methane has a lower specific heat capacity, latent heat of vaporization, and boiling point than water. So we need to go through what's lower than 4.2. Well, that one and that one. Okay. So then what's lower than 2,257? That one and that one. And what's lower than 100? That one. So the answer must be A. In terms of what is common to both RNA and DNA, definitely not thymine, not histones, because RNA doesn't have them. Not deoxyribose, RNA doesn't have that, but both do have nitrogenous bases. Question 8, we've got a graph here which shows that as temperature increases, the rate of respiration increases, and it increases even more if glucose is present. So let's see what conclusions uses lipids. There's nothing about this on lipids. Addition of glucose has a greater impact on rates of cell respiration at lower temperatures. Nope, because the difference there is far less than the difference there. Rates of cell respiration increase with temperature. Yep. More glucose is produced at high temperatures. Nope, because glucose is being used. So C is our answer. Number 9, which wavelength of electromagnetic radiation is absorbed efficiently by chlorophyll? Again, that is just factual recall, I'm afraid, and the answer there was 450 nanometers. In question 10, you're asked to identify what type of fatty acid is shown. So the first thing to look for are the double bonds. and we can see we have double bonds, so it must be unsaturated, so we can cross those off immediately. So then it's a case of, is it cis or is it trans? Well, the hydrogens here are on the same side of the double bond, therefore it is cis and it is B. In question 11, yes, you can't really see the picture, but you don't need to because the picture is irrelevant. It's asking, what does Hershey and Chase conclude from their experiment? And did they include that DNA was mainly outside bacteria? No. Or that viruses infect bacterial cells with proteins? No. That viral DNA was found within bacterial cells? Yes. That neither protein nor DNA chemicals were making up genes in viruses? No. So the answer there was C. In question 12, what you can do here is you can do a quick elimination. So if you go down the role of the DNA primase, does it add a short DNA primer? No. Does it add a short DNA primer? No. Does it add a short RNA primer? Yes. Yes. Okay, so we can get rid of A and B. It replaces RNA with DNA in the Okazaki fragments of the leading strand. Well, the leading strand doesn't have Okazaki fragments. In general, it replaces RNA primers with DNA. Correct. So it is D. Now, question 13 is, I think, a relatively unreasonable question. I haven't really taught you about dideoxyribonucleotides being members of the termination factors because, well, it's a weird little extra fact. I will just go back into the syllabus and double check it. It's factual recall and it's A. I'm sorry about that. I will have another look at that. Question 14, what applies to DNA base sequences? Do some genes not code for proteins? Yes, some genes code for RNA. Are promoters transcribed along with the gene? Absolutely not. Are introns only found within genes coding for proteins? No, introns are found in all genes. Therefore, it is number 1. Now, this diagram is the Krebs cycle, and it's asking you about the number of carbon atoms in Y. So this is citrate, so that's the 6th carbon. It loses a carbon, so that's 5. So we know now that Y is 5 carbons, so we can get rid of B, and we can get rid of D. And then it's how many are the acetyl group? Well, that was 2, wasn't it? Because that was 4 plus 2 equals 6. And so the answer must be A. Question 16 is kind of factual recall, really. What is this? Is rhodopsin found in tears? No. That lowers blood glucose concentration. That doesn't help in blood clotting. So even, I mean, hopefully by elimination, even if you couldn't remember actually what collagen does, you could work that out. Question 17 here shows a diagram in the stage of cell division. Well, there are 2 cells here, so that tells us that it must be meiosis. So we can immediately cross out the 2 that are about mitosis. Now, because we're separating chromatids, it can't be meiosis 1, so it must be meiosis 2, and it is indeed D. Question 18, jumping screen there. Question 18 is asking about cystic fibrosis. Now, cystic fibrosis is a recessive autosomal trait. So it's asking about the genotype of the affected boy's father. So it's this chap and what is his genotype. Now, he is unaffected. Mum is unaffected, but they have children who are affected. So both of them must be big A, little a. So the answer there is C. We messed up with question 19 and gave you 2, but asked you to do the second one. So this is actually a little puzzle to try and solve. So you've got to look at the gene sequences to try and work out where they diverge. So if you go along, you can see that it's the same, and then 1 and 4 diverge off. So that's going to be our first dividing point. Okay, so we're going to have division here, and we're going to have 1 here, and then it divides again because the next base, 1 and 4 are now different, so we're going to have 4 down here, and then that, if we move on, 2 and 3 are the same there, so but they're different from 4, so 2 and 3 are the same but having divided. And so that is the correct answer. Just looking at the pattern of the mutation and of the differences there. And then in question 20, we've got a genetics problem here. So we can see pure breeding plants with purple and long may crossed with pure breeding plants with red and round. So that tells us that purple and long and red and round are our parental phenotypes. This would be a dihybrid heterozygous cross, so you would expect a 9:3 to 3:1 ratio there. Clearly you haven't got that, and what you have fewer of are the purple and rounds and the red and longs, which, as you can see, are the recombinants. So immediately you're saying to yourself, this must be due to linkage. So now let's look at the statements. Purple flowers and long pollen grains are dominant alleles and have assorted independently. No, because we are showing linkage. The genes for flower colour and pollen shape are linked, yes, and all plants producing long pollen grains are recombinants. No. The genes for, so not B. The genes for flower colour and pollen shape are linked, yes, and all plants producing red flowers are recombinants. No, so not C, which leaves us only with D, but let's just check it. Plants producing purple flowers with round pollen grains arose through crossing over. Yes, because purple and round is one of our recombinants, so it must have been formed through crossing over. So the answer there is D.